So in part 2 of this lecture, we'll talk about some of the reasons that we should be careful with the antimicrobials that we do have. So prior to the development of antibiotics, bacteria were incredibly deadly for humans. Bacteria are very efficient pathogens and they kill people very fast. However, in the early part of the 20th century, there were two important discoveries of sulfonamide antibiotics and penicillin. Famously, Alexander Fleming, who discovered penicillin, came back to his laboratory where he had been growing some Staph aureus bacteria and noticed a region of a plate that had been contaminated with some fuzzy-looking mold. Surrounding that mold was a zone where no Staph aureus bacteria were growing. This was the penicillium mold, and the chemical extracted from it, penicillin, saved countless lives in the decades since it has been used. We now have dozens of different penicillin-derived antibiotics that we use regularly in clinical settings. However, within a few short years of the implementation of penicillin into hospitals around the world, many clinicians noticed that infections that used to be susceptible to penicillin treatment were no longer so. Bacteria had developed resistance to the antibiotics. Where do these antibiotic-resistant bacteria come from? Well, it turns out that the principles of natural selection and evolution apply very strongly to bacteria and their development of antibiotic resistance. Let's think about some of Darwin's postulates for natural selection. So first, variation. If we look within natural bacteria populations, there are many mutations within those critical pathways that antibiotics target and work through. And so there are naturally occurring resistances to many of our important antibiotics. Also, those genes are heritable. They can be passed from generation to generation when those microbes reproduce. And also importantly for bacteria, some of those genes can be passed even to other species of bacteria through horizontal gene transfer. And finally, because antibiotics have become so effective at fighting bacteria in a variety of different settings, we've over-implemented and begun to use those antibiotics all over the world. We now use antibiotics not only in medicine, but in farming, industry, and also have begun to overprescribe antibiotics in clinical situations where it's not appropriate for an antibiotic to be used. This graphic shows how antibiotic-resistant bacteria might spread throughout the world. So, remember variation. There are lots of bacteria, and antibiotics are broadly effective against most of them. But within any bacterial population, there are genetic variants, mutations, or bacteria within an environment that naturally have resistance to that antibiotic. Now, in any situation where we use antibiotics, those antibiotics will kill bacteria that are susceptible. Some of those bacteria will become, that are resistant, then are left behind. Once all the competing bacteria have been killed by the antibiotic, the resistant bacteria are able to grow, use the resources available in the environment, and reproduce to the point where they are now dominant in the population. Those resistant bacteria can spread to multiple locations, and occasionally they can pass their drug-resistant genes to other bacteria, causing more problems. So antibiotic-resistant bacteria have spread around the planet because of our overuse of antibiotics. For example, one of the widest uses of antibiotics is in farm animals. We commonly give animals that are raised for their meat antibiotics because it increases the rate that they grow. Therefore, they produce more meat to sell in the market. However, those animals have a variety of different important pathogens in their microbiota. Those antibiotic-resistant bacteria can become more prominent, then they can be present on the meat when it's sold in the store. If that meat isn't cooked or handled properly, people can become sick with those antibiotic-resistant bacteria. Also, the antibiotics are secreted from the animals in their urine and feces. They can wash into a variety of different environments from farm runoff. That allows the development of antibiotic-resistant bacteria throughout the environment that can then come into contact with people. We've also seen the overuse of antibiotics in medical settings. For example, we know that infections can be caused by a variety of different pathogens, including viruses, fungi, and bacteria. Our medications, our antivirals and antifungals, aren't typically as useful as antibiotics. And so, in some cases, when a person comes into a clinic complaining of disease symptoms, we don't necessarily know whether or not their infection is caused by a bacteria or a virus or a fungi. The only one of those three that we have an easily deliverable medication, though, for is the bacterial infections. And so in many cases, people who want treatment for a condition might be prescribed antibiotics, even though they aren't appropriate to be used in their particular disease. Those antibiotics can still affect the bacteria that are present in a person's microbiota, and that allows some of the antibiotic-resistant microbiota to become more prevalent, and those microbiota can occasionally cause diseases. Bacteria are able to resist the effects of antibiotics through a variety of different mechanisms. One, some bacteria develop mechanisms of drug modification or inactivation, and this is where the bacteria have enzymes that chemically modify the drugs and make them ineffective. Bacteria can also prevent the cells from uptaking those antibiotics by blocking their entry into the cell. And they also can produce efflux pumps, which increase the removal of those drugs from the cell. So efflux pumps are transmembrane proteins that remove antibiotic drugs from inside the cell and prevent the cell from being harmed by them. Also, mutations in the bacteria to the target of the antibiotic. So remember that antibiotics target specific pathways or specific biochemical processes that are critical for the cell. Those enzymes or biochemical processes can be modified. And so the target modification, the target of the antibiotic, can mutate and prevent the antibiotic from being used. And we're going to see an example of that in the next few slides. There are also a couple other mechanisms of antibiotic resistance that some bacteria develop, but those first three are the most important ones. So one specific strain of antibiotic-resistant bacteria that's become problematic in modern medicine is MRSA, methicillin-resistant Staphylococcus aureus. So Staph aureus is a gram-positive part of the normal microbiota. You remember that we discussed Staph aureus a few lectures ago, and this is present on many people's skin. It can cause skin infections, and it can cause more serious infections if it gets introduced into sensitive areas of the body. So as a gram-positive, Staph aureus are generally susceptible to penicillins. In fact, penicillins were first discovered because of their effect on Staph aureus bacteria. Penicillins are broad-spectrum beta-lactam antibiotics, and beta-lactam antibiotics work by preventing bacteria from forming their cell walls. You'll remember that gram-positive bacteria have very thick cell walls, and these are important parts of their cell physiology. However, we did notice that resistant strains of Staphylococcus aureus developed within just a few short years of implementing their use of penicillins in the clinic. And that's because Staphylococci bacteria, many strains of these intrinsically produce beta-lactamase, which is an enzyme that breaks down the beta-lactam chemical ring and prevents these antibiotics from working. This is a mechanism of drug modification. So once that beta-lactam ring is broken, the penicillin is no longer effective. So the drug methicillin was developed as a chemical derivative of penicillin. It was modified in a chemistry lab in order to prevent the effects of the beta-lactamase. So methicillin is a drug that's able to still prevent the bacteria from forming their cell walls, but it's not affected by the beta-lactamase enzyme that breaks down the antibiotic. However, methicillin-resistant Staphylococcus aureus strains began appearing in hospitals around the world in the 1960s. To understand how this MRSA bacteria became so widespread, we first have to understand a little bit more specifically how methicillin works. So penicillins are competitive inhibitors. Remember, those are enzyme inhibitors that work by blocking the active site of an enzyme. The enzyme that penicillins block is an enzyme called penicillin binding protein. It's named that because we discovered it because it was the target of this important antibiotic. Penicillin binding protein is actually responsible for, in the bacteria, forming chemical crosslinks between the peptide parts of the peptoglycan. Remember, those peptides form these important crosslinks between the rows of the carbohydrate NAG-NAM chains. So in the bacteria cell wall, we see that penicillin binding protein is important for forming those chemical bonds, and penicillin and its derivatives, including moxicillin and methicillin, all work by preventing the formation of those chemical bonds. So antibiotic sensitive Staphylococcus aureus, bacteria that are affected by those penicillin antibiotics, they have an active site that is able to chemically catalyze the formation of those peptide crosslinks. And the antibiotic methicillin and its derivatives are able to fit into that active site and block the chemistry from happening. However, antibiotic resistant or methicillin resistant Staph aureus have a change, a different chemical shape of that active site that happens because of an amino acid change within that protein enzyme. So that amino acid mutation allows that enzyme to still catalyze the chemical crosslink, but the antibiotic itself no longer fits into the active site. And so even though the enzyme is still able to carry out its job in the cell, the antibiotic is no longer able to block it. So we do have backup antibiotics to treat MRSA infections. If a Staph aureus infection is resistant to methicillin, the antibiotic vancomycin can be used. However, vancomycin is an extremely has a significant toxicity to people who take it. Vancomycin can only be delivered intravenously in a hospital, and so it's rather expensive to give this drug, and also it causes significant kidney damage. And so people can only take vancomycin for a short period of time without experiencing some of those toxic side effects. MRSA is also problematic in healthcare settings because Staph aureus in general are carried on many people's skin and also form biofilms, which are resistant to many of our sanitation and disinfection processes that we use on hospital items. And so we need to take many steps in order to prevent the spread of MRSA bacteria. We need to identify people that may have natural or that may have MRSA as part of their normal microbiota, because they can potentially spread the bacteria to others. Patients who do have MRSA infections should have limited contact with other patients. Remember, the hospital is a setting where many sick people are kept in a very small area. And so serious diseases that some of those people have could spread to other patients and make them even more sick. We also should be careful with our use of the antibiotics that we do have and prevent their misuse so that those antibiotic-resistant bacteria don't spread. So antibiotic resistance doesn't just develop because of our misuse of antibiotics. Some individuals, such as individuals with a genetic condition called cystic fibrosis, develop antibiotic-resistant microbiota through no fault of their own. So cystic fibrosis, or CF, is a genetic disorder, and it's caused by a mutation in the gene that codes for the human protein CFTR. What CFTR does is it regulates the passage of ions in and out of the human cell membrane. And so along the epithelium, which is that layer of cells that lines the areas of our body open to the outside, including our airways, CFTR is normally important for controlling osmosis because it regulates the passage of ions in and out of the cell. It also controls the movement of water in and out of the cell. So people with mutations in CFTR that prevent that transmembrane protein from transporting ions in and out of the cell develop a thick layer of mucus along their epithelium. So this mucus tends to clog airways, and it provides an environment for many bacteria to become trapped within the airways and multiply. And this means that CF patients develop frequent respiratory infections by the bacteria that are trapped in that thick layer of mucus. So our tools to fight those respiratory bacterial infections are antibiotics. And so people with CF have to frequently go into the hospital and be treated with antibiotics. But because they're frequently being exposed to antibiotics, the bacteria in that person's airways, some mutants in those bacterial populations are resistant to those antibiotics. And those resistant bacteria will become more populated in that CF patient's microbiota. And eventually, CF patients develop chronic infections of bacteria that are completely resistant to all of the available antibiotics. And because of this, many of them will eventually succumb to those infections. And the life expectancy of somebody with CF is around 48 years. So in CF patients, we see that even though antibiotics temporarily get rid of the infection, those infectious bacteria then repopulate because not all are killed by the antibiotic. So this is a reinfection by resistant bacteria. So the bacteria that are left behind that aren't killed by the antibiotics have mutations that make them resistant to the antibiotics, and that means that those bacteria are going to be resistant to the antibiotic, and it's not going to be an effective treatment the second time that it is applied. However, some reinfections, where an infection comes back after an antibiotic treatment appears to work, are not caused by antibiotic-resistant bacteria. They're caused by persistent bacteria. Persisters are bacteria cells that survive antibiotics by going dormant. So if we look at a population of bacteria cells, even though they are genetically all the same, some of those bacteria cells have this reduced metabolism where they're not using energy as fast and they're not moving chemicals in and out of the cell quite as fast. And that means that even though an antibiotic is applied, those cells aren't killed by the antibiotic. So in any infection, it's necessary for the person's immune system to clear those last few cells from the body. Occasionally that doesn't happen, and when those persister cells remain, then they can, once the antibiotic is removed, they can repopulate and that infection can recur. However, an important difference between reinfection by persisters and reinfection by resistant bacteria is that those persistent bacteria, the infection is still going to be susceptible to the antibiotic. And so this means that we need to identify whether or not a reinfection is caused by resistant bacteria or bacteria that are still sensitive to the antibiotic, because that can determine whether or not a treatment may or may not still work. So in order to develop new antibiotics, we need to have a way to test to see if those antibiotics work and if they are safe, free enough from side effects to implement in the clinic. So an important set of experiments that we do to trust all new medications in the United States are clinical trials. In the United States, these trials are approved by the Food and Drug Administration, or FDA, which has new clinical trial designs are submitted by pharmaceutical manufacturers and developers to the FDA, and the FDA independently monitors those trials and determines whether or not the data is sufficient to warrant the implementation of a drug into the market. Clinical trials are expensive and carefully done because we want to know whether or not the drugs that we are using in medical settings are safe and effective. And so clinical trials are some of the biggest and most important experiments that we do in modern science. There are 3 phases of clinical trials. Each phase has a different goal, a different outcome of the experiment that the clinical trial administrators are looking for. In phase 1, this is the first phase where the drug is moved into and studied in people. The goal of this phase of the trial is safety. Determine whether or not the drug is safe and at what dose it's safe at. So a small number of subjects are given the potential drug and in increasing concentrations. And that means that they're increasing the amount of the drug that they're giving the patients in that phase of the trial to determine what dose of the drug is safe. As long as no serious side effects are identified, and as long as a safe dosage of the drug can be determined, then that drug moves on to phase 2. In phase 2, a larger number of subjects are given the drug or a placebo. And so some of the individuals in the trial receive the actual medication, and some of the individuals in the trial instead receive a fake medication, a placebo. This allows people in both experimental groups to be unaware about whether or not they're receiving a real or fake drug. And it allows the effectiveness or the efficacy of the drug to be determined as the amount of patients who do better in the drug group compared to the placebo group. In phase 3 of the trial, the drug is tested in a larger number of subjects. Typically, for new antibiotics, the drug is not measured against a placebo, but against existing therapies. So some people get the new medication, some people in the trial get a medication that's already been approved and is effective at treating the disease in a large population. And that means in order to bring a new drug to market, it has to offer some benefit compared to drugs that are already on the market. So this is a very high standard, and each one of these phases determines whether or not the drug is safe and whether or not it's actually worthwhile for people to be given the drug in a clinical setting.